Hello and a very warm welcome to you all, dear students. In my last lecture, I had discussed about mutations. Now we go one step further and try to see the types of mutations. Mutation, as we saw, has been defined in various ways, in various terminologies. There are different perspectives of mutations. National Human Genome Research Institute puts a very interesting definition of mutation. In one line, they say that any change in the sequence of DNA is mutation. The simplest form of definition of a mutation can be any change in the sequence of DNA. Where does this affect? We know it affects the protein synthesis. Mutations, we have seen, they are those heritable changes, permanent changes, sudden changes which have a marked effect on the phenotype, can be called as mutations. Mutations, they have a huge, what you can say, impact on not only the phenotypic characters but also the genotypic characters and the survival of organism. Having said that, not only the survival of the organism for one generation, but generations to generation, up to such a great extent that time may arrive in this process where the cell may be called as evolved into a new character. So mutation is a broad, a huge concept which cannot be summarized in one lecture or two. But here, interestingly, we will have a perspective of a prokaryotic mutation, single celled mutation. In the process, we will also see what it happens when there are mutations in the chromosome, and also we will try to draw parallels with the different effects which this mutation has on the DNA. So the central dogma you always have to remember whenever I say or talk anything about genetics, especially the part where a protein is synthesized. In prokaryotes, this is a remarkable and unique phenomenon that transcription and translation, they are coupled. I think that this is a primitive process which has not just evolved, means they resisted the change and they just lived as they lived some thousands of years ago. And this process of coupled transcription and translation is directly affected by, in process, I can say in vivo, in process, it is directly affected if there is any change on the base sequence on the DNA. DNA, it has a very natural type of Watson and Crick pairing, adenine pairs with thymine, guanine pairs with cytosine. There are millions of bases running along the ladder or the strung of the DNA, and each and every cell, each and every individual living system on this earth has its own unique copy of a DNA. And what is that uniqueness? That uniqueness is the arrangement of nitrogen bases on the DNA. That is what dictates everything in the life of that cell, of that tissue, of that organ, of that organism as such. So when there is a change, now there are various aspects to study when I say when there is a change on the DNA. So how does this change occur? Why does this change occur? What impact this change has? All these things we are going to see in this and in subsequent lectures, next lectures. Today I will be mainly focusing on the types of the DNA. So when there is a change, we call a sudden permanent change on the DNA. Now you see, when we have a observation or empirical data that 1000 base pairs per second the DNA polymerase copies down the DNA, duplicates the DNA, and there are randomly incorporated bases which are wrong. Randomly, wrongly incorporated base pairs are very frequent, but evolution has created two systems in DNA replication. One is methylation, methylated bases, which are the markers, which are the, what you can say, identity batches of that cell, which does not allow any mistake to occur. and second is the editing function or the proofreading function of the DNA polymerase itself, where it does not allow any mistakes to occur. So it is a foolproof process. DNA replication is a foolproof process, due to which, try to understand, the rate of mutation in any cell is very low, as low as we have seen 10 raised to 6 to 10 raised to 12 is the rate of mutation. I mean, one cell in 10 raised to 6 cell may undergo a permanent change. Now this rate still is there. How does it happen? How does this sudden permanent change it occurs? Now there are two perspectives to study that, which I will be covering in my next lecture. That is are induced mutation and spontaneous mutation. That is in the next slide. But here just see what we are talking about when we are talking about mutations. We are talking about variation, observable and a non-observable change in that system, in that life system, which is attributed to DNA. So we will call it as a genotypic and phenotypic variation. What are these genotypic and phenotypic variations? Genotype, when you say, is the constitution of a cell that is transmitted to its progeny. All the genetic information transmitted from a parent to a progeny is the genotype. And phenotype is all the physical observable thing, like maybe the height, or if you are talking about a bacterial cell, all the colony characters which are there. Size, shape, morphology, elevation, all the 10 to 12 characters which you see, that is phenotypic. Genotypic is whether that has the gene for flagella, whether that is a gene for spore formation, whether that has a gene for motility, so on and so forth. Whether it has a gene for synthesis of tryptophan, whether that has a gene for utilization of lactose. Bacterial genotypic characters. Phenotypic of a bacteria, all the 10 colony characters. If you see the multicellular system, you see this figure, the tigers, these 4 tigers, they are having a genotypic, maybe we are not knowing which is a ferocious tiger and which is a very mild tiger. It is not in always attacking mode. It can be tamed. Out of this one cannot be tamed. It has its wild character. Maybe out of this one can be tamed. I am just guessing. Okay, we don't know. Those are the genotypic characters, and the phenotypic characters is what you see. One is white, one is a mix of white and the original wild type striped tiger. This is somewhere a cross between a non-striped and a purely striped. This is somewhere in the middle. So this is phenotypic variation. This exists in all living systems, that is plants, animals, human beings, unicellular systems, all types of, on the land, in the sea, in the air, birds, everywhere. One of the basic purpose of studying prokaryotes is to understand eukaryotes. Try to understand this. When you study genetics of prokaryotes, our focus, our aim should be to understand the multicellular system, because studying one bacteria, which is a remarkable model of the multicellular system, serves the purpose to understand the multicellular system. Not only that much, today we have crossed boundaries of using these unicellular genetic systems into multicellular system, like production of insulin using E. coli for humans. This simple example sums it all. How the genetic systems, they are similar across the boundaries of life. That is what is said when we say genetic code is universal in nature. So when this genetic code expresses, we have a genotypic and phenotypic variations. I am like my father, but I am not like my father, isn't it? You are just a, what you can say, not a clone, but have most of the characters attributed from your mother and father, but still you are not 100% same as mother and father. That is the genotypic and the phenotypic variations. Okay, variations, the phenotypic variations are influenced by the environment. They are temporary and not heritable, that is the variations which are impacted by the environment on the phenotype. Variations, when I say here, this is the environmental variations and hereditary variations. And hereditary variations, they are influenced and they are transmitted from one generation to the another generation. Now, what are the mechanisms of these genetic variations? Why do you have genetic variations when one character from one progeny or one parent is transferred to its progeny, you have a genetic variation. When it is natural, it is just because of mitosis, meiosis and mixing of the genes in the eukaryotes. But in prokaryotes, we have studied three different mechanisms of genetic variations which are not mutations. Try to understand. Here when I am talking about mechanism of genetic variation, I don't really mean mutation, but mutation is just an example of genetic variation. And what is not mutation but still a genetic variation is transformation, transduction and conjugation. We have studied this earlier in your last semester, that transfer of or exchange of genetic material through transformation, conjugation, transposition, all these where there is some agency mediating the transfer and a new trait is accepted by the recipient cell. This is genetic variation, it is not mutation. Means the transfer which results into a change in the phenotype and the genotype, but still not via mutation, but a very natural phenomenon. Just like we are a product of our parents' mixing of genes, which took place in the sperm and the egg level. Mitosis and meiosis, continuous shuffling, mixing, genetic exchange between the chromosomal, that is, we call it as variations, took place, and you are what you are today, a perfect breed of your mother and father. So that is change seen but not mutation. Now the second example of mechanism of genetic variation is mutation. Mutation, we have defined mutation, a change in the DNA sequence which alters the change in phenotype and genotype. Now mutation can be spontaneous or induced in nature. This topic of spontaneous and induced mutation and its mechanism I will be delivering in my next lecture. Today we are here to study the types of mutation. When a mutation occurs by spontaneous mode, spontaneous is naturally which is occurring over the timeline of evolution, and induced is the one where in a particular generation physical and chemical agents, like cancer is one mutation in humans, it is induced. There are n number of factors. You cannot say that this particular thing has induced a cancer. No, even a simple diet where free radicals are formed and complete oxidation products are releasing more free radicals may also result into cancer. We say that he was nice, he doesn't have any bad habits, still he is having cancer. Well, quite possible that is. That is induced mutation. In bacteria, induced mutations results into different types of characters, prokaryotic traits, which we will see. So these are called as genetic variation, and one of the mechanism of genetic variation is mutation, which we will be seeing in detail today. Now, categorizing mutation. If I ask you a question, how many types of mutations are there? This is a very huge question. It cannot be encompassed in one single line answer. What is your aim of asking me how many types of mutations are there? My counter question will be, are you talking in eukaryotes or are you talking in prokaryotes? Means in a multicellular system where there is a huge chromosome, a very complicated structure, wound around histone proteins arranged systematically, a set of chromosomes X and Y, in female XX and XY in males. In those systems, and that too a sperm fusing with a egg, totipotency, the germline, there when they grow, and continuous crossing over of the alleles and the segments, and selection of genes, that is the dominant and the wild genes, is being done for 9 months inside a baby to create a complete character. 9 months for a baby inside a womb to create a complete character. This mutation is totally different from the prokaryotic mutation where only one single bacterial cell is there. So if you just categorize mutations in multicellular systems and mutations in prokaryotic system, I would like to say in eukaryotes chromosomal mutations are there, and copy number variation also is a type of mutation. Copy number 24, you say chromosomal number. If there is variation, even in the number of genes in that chromosomes, if there is variation, then that results into a mutant genotype or a phenotype. So chromosomal mutation. In chromosomal mutation, if you see, inversion, deletion, duplication, translocation, these are the types of mutation in multicellular system. Inversion is where one region of a chromosome is flipped and reinserted. If you see the antibody structure, it is a very complex structure. It has arms. It is a product of some gene. Okay, the gene which is present on a chromosome somewhere, if that is flipped or reinserted or something happens, like it happens in transposition, then there is an inversion of a gene. That is called as an inversion. The whole gene is inverted. Deletion, a region of a chromosome is completely lost, resulting in absence of all the genes in that area. We are talking about eukaryotes, multicellular system. We call this DNA as a large DNA as compared to the small, compact, covalently closed circular DNA of bacteria. So deletion of a region of a chromosome, resulting in the absence of, literally absence of that gene. Most of the genetically inherited diseases are associated with deletion. Duplication, a region of a chromosome is duplicated, resulting in an increase of the dosage of genes in that region. So some people, they have 6 fingers, so extra finger. There is a duplication of the gene. So duplication is being observed in a phenotype. Same thing may happen in the internal genetic systems also. Okay, duplication. Translocation, a region from one chromosome is aberrantly attached to another chromosome. It is a huge system. A set of chromosomes are there. We can see this in yeast also, in humans, animals. You have many abnormalities in the birth. Okay, so what is this happening? A region of one chromosome, that is one whole region, that way set of genes is aberrantly attached to the another chromosome. Translocation of the genes. So these are chromosomal mutations. Of course, I am just mentioning you to make the picture clear that this is the type of class of mutation, the type and what happens in eukaryotes. The second class of mutation in eukaryotes is copy number variation. They have copies. In bacteria, only one DNA is there. In multicellular system, they have copies. So what happens with that copy? If the copy number is enhanced, 24, you have extra number of chromosome, gene amplification. Number of copies of a locus is increased. Number of tandem copies, means one behind the another, they suddenly increase. Gene amplification. Then expanding trinucleotide repeats. The normal number of repeated trinucleotide sequences which are present, trinucleotide sequences, they are suddenly repeated. So you have a sudden extra copy of that region of the locus. So we are talking about a gene segments which are showing variations. See, this is variation, gene amplification, expanding the trinucleotide region. This is all involved in the copy number variation. And all this results into vital, lethal, supervital types of mutations. So this is in eukaryotes. The whole scenario becomes very simple, but try to understand whatever this has, the effect on the mutation, the effect of the mutation on the metabolism is same in both. What is same? They are markedly affecting the protein synthesis. A change in the DNA sequence alters the change in the mRNA, which alters the change of the amino acid sequence in the protein. This is basic. Okay, in both eukaryotes and prokaryotes. So let us move on to the eukaryotes to see. It's very simple. We have one single type or class of mutation in bacteria, that is called as point mutation. Whatever the mutations you are studying, all will come under point mutation. Now this point mutation has 3 classes, subclasses, or you can say 3 different styles of functioning. Let us call them as types of mutation. Point mutation can be substitution, insertion, deletion. The name itself in English sums it all. Substitution. Let us imagine 3 of your friends are sitting on one bench. One student is being replaced by another. Means there are, for example, 3 students and one is replaced by, substituted. Insertion is adding one student in them, that will become 4, or the sequence will be changed. Deletion is removing one student, so you have 2, the next bench will be considered the third base. So substitution, insertion and deletions, now their impact, or what are the styles of substitution, what does it has, this will be further, these types of mutation, I will explain what happens, are only 3: substitution, insertion and deletion, and their effects which are there. The effect which these 3 types of mutation have on the genetic system or the functioning of the gene, that is protein synthesis, is it can be silent mutation, sense mutation, nonsense mutation, missense mutation, frame shift mutations, additions, deletions, multiple types of effects or style of functioning is there. That will be what my next slides will tell you, how these 3 types of mutations function. Okay, so whatever the mutation is there in prokaryote, that is point mutation. 3 types of point mutations are there: substitution of a base, where one base is incorrectly added during replication and it replaces the pair in the complementary strand. So substitution. So AT base pair gets converted into GC base pair, or a GC base pair gets converted into AT base pair. That we will see. Insertion. So if there is AAA, a T is suddenly inserted. What will happen? It will be ATA. So one or more extra nucleotides are inserted into a replicated DNA. So this results in the shift in the frame. This is called as frame shift mutation. If this frame shift mutation results into a nonsense codon, it is called as a nonsense mutation. So try to understand the hierarchy or the sequential categorization of this mutation. Basically, you have to remember it is point mutation in bacteria, and 3 types are there: substitution, insertion and deletion. Finish. Substitution results in frame shifts. Insertion and deletions also result in frame shifts. We will see that as in the examples. Deletion is where one or more nucleotides, they are skipped or not added or deleted during the replication process, and this also results in a complete change in the reading frame. This is called as frame shift mutation. So let us just see these point mutations. These are silent, nonsense and missense. Now point mutations, if you see here, I said point mutation, substitution, insertion and deletion. You have to remember this very perfectly: substitution, insertion and deletion. And these 3 may result into silent, nonsense or missense mutation. There can be some change and there may not be any mutations also. This is no mutation. Means a change in the base but no mutation. That is a silent mutation. Nor no mutation at all. Means A is replaced by analog of A, which doesn't have any marked effect on the phenotype. Okay. So what is a point mutation? Point mutations in bacteria, as it is now, you should have understood, is only category of mutations in prokaryotes, which are such a large category of mutations which describe a change in single nucleotide of a DNA, such that nucleotide which is there, that is switched to another nucleotide. It becomes another nucleotide by addition, deletion, substitution, whatever it may be. One base is switched for another nucleotide. It may be deleted or it may be added, which causes the DNA to be totally different from what it was being earlier. The earlier we will call it as a wild type DNA, and this changed DNA we will call it as a mutant DNA. Wild type DNA and a mutated DNA. Okay. So you see the silent mutation. Now this is a DNA level where TTC is there. This C is replaced by T. So which was coding for lysine, now it also codes for lysine. Why? Because degeneracy of the code TTC and TTT, they are same, they are coding for lysine. So such type of mutation which does not alter the phenotype of a protein are called as silent mutations. Now what happens, you see, if this first T is replaced by A in the DNA level, first T is replaced by A. Now on the mRNA, what is happening? In the earlier example, TTC resulted into AAG on the mRNA, resulted into AAA on the mRNA. AAG and AAA are redundant codons. Redundant means there are 2 different codons coding for the same amino acid. Therefore, it becomes a silent mutation. Now in the first same example TTC, if first T is replaced by A, what will happen at the mRNA level? This A will add UAG and it becomes a stop codon. In the middle of the reading frame, if this is happening, this will suddenly result into a nonsense codon. Any three of UAG, UGA or UAA, if this results due to a point mutation, insertion, deletion, substitution, whatever it may be, then it is called as a nonsense mutation. So remember, a nonsense mutation always forms nonsense codons, termination codons, and it results into stopping the protein synthesis abruptly, randomly. Now there is one more, you can see here, TTC, in the middle it is changing into C, or in the middle it is changing into G, which changes the code into AGG or ACG, which instead of coding for lysine, they are coding for arginine and threonine. So what is happening? If these are the prime amino acids in the active site of the protein, then it will make no sense, and it will also alter the phenotype. So these are the three types of point mutations. Now this point mutation, the same, if you see, it is random, undirected, heritable variation by a change in the nucleotide base sequence in the DNA. Okay, what is point mutation? It is nothing but the mutation in the bacteria which are random, undirected, heritable variations caused by a change in the nucleotide base sequence of the DNA. So this UAA, as you can see, it can result into silent mutation, missense mutation, nonsense mutation, as I just given you the examples. Now what are the effects of this point mutation? This effect of this point mutation on the mRNA or the mRNA which will translate it into protein, it can result into frame shift mutation or base pair substitution. There are two basic effects of mutations, point mutations, that it can result into a change in the reading frame, frame shift mutation, where a complete sequence is changed and a complete protein is altered, whereas base pair substitution is resulting into either of this one or sequentially anything in the middle, silent mutation, missense mutation, nonsense mutation. Silent mutation is change in the codon but no change in the amino acid. Missense codon is change in the amino acid which may or may not alter the protein. If it is an important one in the, let us say valine is there, and it has the same hydrophobic characters as that of the original one, leucine, then, and it is not having any change in the protein structure, it can be missense. And nonsense is which is resulting into premature termination of the protein synthesis. Now why or how does this mutation function? It functions due to mutagenic agents. This is something which we will be covering in my next lecture, so I will not deal with that here right now. We will just focus on the types of mutation. So let us see the first type of point mutation, that is frame shift mutation, where addition or deletion of a base takes place. Addition or deletion is an event which changes all the codons downstream from the position onwards of that change, wherever that has changed. Now you see, this is TAT, TCG, CTA, GTA, CAT, and this is on the mRNA. We will directly see the effect on the mRNA. Of course, this has a change on the DNA, due to which there is a change on the mRNA. So there suddenly, instead of this G, we have whatever it may be, addition or deletion. Let's see, in between this T and G there is addition of C. Between thymine and guanine, this is not G not being replaced by C, but in between T and G there is C. So suddenly TCG, and the second will be GCT. You can see GCT. If there is, see the red laser pointer here, in between T and G, if there is cytosine added, suddenly the next codon will be TCG, then GCT, then again AGT and ACA. See here, and this results into complete change in the reading frame. So that they are called as frame shift mutation. Same thing happens if a base is deleted. in the end of this lecture, I will give you some animated examples regarding this. Here you just understand through this slide that deletion now, GAA, GCA, GTG, if G is deleted, the next codon will be AAG. Because the code is commaless, it is not having any full stops, it is a commaless code, it is a non-overlapping code. So if you remove this, suddenly the next three bases will form a pair. So AAG will be the next one, CAG will be the next one, GT, so on and so forth. What that happens? So if GAA was coding for glutamine, then AAG will code for lysine. If GCA was coding for alanine, then this CAC will code for histidine. So there is a complete shift in the reading frame. This is the effect of addition or deletion, which is a class of point mutation. As it results into a shift in the reading frame, it is called as frame shift mutations. Now the same, you can see some examples. You can stop this lecture here, you can pause for some time, go through the example. The same point mutation, substitution of a single base, silent mutation which has no effect on the protein synthesis. As you can see here at this particular point here, the wild type DNA which is there, AGC, this sequence AGC, TA, this A is replaced by T, and due to which what is happening on the mRNA, GUA is converted into GUU, but valine is substituted for valine, because GUU also codes for valine, GUA also codes for valine. This is due to the redundancy of the code, degeneracy of the code. The code is degenerate. Okay, so that is why no change point mutation, which has silent effect. It is called as a silent mutation. Second is a missense mutation, where it results in amino acid substitution. Okay, so here you can see GUA is there, GUA, and next CCC, which was for proline, has converted. There is a addition or whatever it may be, substitution of a base. ACC results into threonine. So it results in amino acid change, only change in one amino acid. Now whether that amino acid will have a marked effect on the phenotype or not, that depends upon its function in the protein. There is a huge number of amino acid required for the protein folding, and only a small amount of amino acids inside that folding which acts as active sites. If that amino acid is somewhere outside, it is only for the structural formation, not the active site formation, then this point mutation will not have any marked effect on the phenotype. But if it is in the active site, then it will have a marked effect. So point mutations, you cannot say whether that is really lethal or not lethal. Depends upon where one single amino acid change is present in the structure of the protein. So that is called as a missense mutation. Then substitution of a single base resulting into a stop codon. So TAG is, the C is substituted by G here. So this UAC got converted into UAG. Tyrosine, which was a functional amino acid, got converted into a nonsense codon where there is no amino acid, no tRNA. Protein synthesis stops here. Therefore, it is called as a nonsense mutation. Any mutation which substitutes a functional codon to a stop codon or the termination codon, that is called as a nonsense mutation. Frame shift mutations are those mutations where addition or deletion of a base takes place, which results into a complete shift in the reading frame, as we saw in the last example. Same thing here. As you can say, TA is suddenly removed here. This TA which is there, this is a normal DNA, and TA is removed. So as TA is removed, you have AGCGC. So AGC is one codon, then you can see the laser pointer, GCC will become the second codon. You see here, GCC becomes the second codon. And as there is a change in the reading frame completely, you see all the sequences of amino acids are changed. So by far, frame shift mutations have the most deleterious effect. That is a marked effect which is towards lethality, non-functionality, or super vitality. This may result into a super character also, but mostly it is observed that for a particular generation it has a lethal effect on the phenotype. So this is frame shift mutation. So lastly, base pair substitution, the last type of mutation which is there, base pair substitution. These base pair substitutions are a result of transition and transversion, maybe one of the reasons. What is transition? When a purine base is replaced by a purine base. Adenine and guanine, these are purines. Cytosine and thymine, these are pyrimidines. Okay, adenine and guanine, purines. Cytosine and thymine, they are pyrimidines. So this purine, double ring structure, pyrimidine, single ring structure, purines and pyrimidines, adenine, guanine, cytosine and thymine. So what happens is that when a purine is replaced by a purine, or a pyrimidine is replaced by a pyrimidine, out of these four bases which are there, purine being replaced by a purine, pyrimidine replaced by a pyrimidine, is called as transition. Very simple as that. And transversion is alternate. If a, see here, transversions, okay, these are transitions, adenine, guanine, purine to a purine, cytosine replaced by thymine or thymine replaced by cytosine, transition. Adenine and cytosine, transversion. Guanine and thymine, guanine replaced by thymine, thymine replaced by guanine, transversions. Okay, guanine replaced by cytosine, transversion. Thymine replaced by adenine, that is transversion. Okay, so there are 4 types of transversion possible and 2 types of transitions possible. You can see here, when this happens, when a T is, let us see here, C is replaced by G. What type of is this? C. The C is replaced by G. So this is a transversion type of substitution, and this has a complete change in the reading frame. So this is one type of point mutation in bacteria. Now we will see, this is the almost what you can say, the same example given here, base pair substitution. I would like to mention here, base pair substitutions are also called as switches, because substitution mutation just switch out one base, one or more base for a different one, means they are not adding, they are not deleting, but they are substituting, changing. You see UAA, the first U has been substituted for C. Okay, C. Or you can see here, the first U has been substituted for G. So there is a substitution. It is a mutation which exchanges one base for another. That is a change in single nucleotide, such as switching A into G. Okay, here such a substitution could change a codon into one codon that is different from the amino acid. So what it will have? It will have the same effect, that is silent, missense, nonsense. So this silent mutation, missense mutation, nonsense mutation, you can say, are the effect of the mutational process. And base pair substitution, frame shift mutation, they are the types of point mutation. Many of these substitute usually into an incorrect amino acid in the corresponding position of the protein, and when this happens in large proportion, the protein function is markedly affected, it is altered. Some base pairs, when they produce a stop codon, a premature, it is a speed breaker, halting of the ribosome takes place, everything dissociates, and premature ending of the protein synthesis takes place. So these are all the types of mutations. These are the basic types of mutation. Now, originally in nature, if you see what causes this mutation, there are 2 basic reasons on the molecular level. Now this is the types. What causes, what is the molecular explanation for this mutation? The molecular explanation for this mutation is spontaneous mutation and induced mutation. In nature, whatever is happening without the effect of the environment is not playing, but due to continuous copying down, continuous one mistake sustains in 1000 generations, 2000 generations, 10,000 generations, 100,000 generations, one marked phenotypic or genotypic character is changed spontaneously without any reason, but still it has a molecular explanation. And induced are those which, for a particular generation, when there is ultraviolet radiation or chemical mutagenic agents, or there are base analogous compounds, there are acridine dyes, There are many things, many chemical and physical agents which induce a change in the DNA. You are eating so much of food, you are breathing so much of polluted air, your lifestyle is somewhat which is prone to exposing your DNA to large junk of chemicals which may induce change in the DNA. Junk food is there, carcinogenic compounds are there, colored foods which are adding color additives, huge chances of induced mutation. But that induced mutation is acting only on that generation, not for the next, not that fast. Okay, if that sustains in the next generation, we call it as a hereditary disease or a heritable change. All this we will see in my next lecture. But before ending this, just let's see the mutation, frame shift, addition, deletion and substitution in animated form in 5 minutes, in a very short duration. Come, let's see the whole process revised in animation. And in my next lecture, I will be talking about spontaneous mutation and induced mutation. Also, I request you all to see the links given below for further studying and notes preparation. So let's go and see, have a look at the animated version of these mutation types. So let's see the addition and deletion type of mutations here. If you see the screen, the nucleotide sequence in DNA determines the nucleotide sequence in messenger RNA and consequently the sequence of amino acid in a protein. A mutation in the DNA can result in a change in the amino acid sequence of the protein. One type of mutation which can occur either spontaneously or as a result of mutagen is the addition of one or more nucleotides during the DNA replication. You can see here, because translation of a gene begins with a specific codon and it proceeds one codon at a time, the addition of an extra nucleotide, it shifts the codon in the mRNA. This type of mutation is called as frame shift mutation. A frame shift mutation affects all the amino acids incorporated beyond the original protein. If a new codon is generated by frame shift, is a stop codon, then the protein synthesized will be shortened and it will often be non-functional in nature. Another type of frame shift mutation, as you can see here, is when nucleotide is deleted during the DNA replication. Deletion of this nucleotide in DNA results in a change in the codon in messenger RNA from the point of deletion and it changes the amino acid inserted into that protein. So this is the effect of deletion of a nucleotide. So we are calling it as addition and deletion type of mutation. Let us now see the base pair substitution type of. So in a mutation by a base pair substitution, a mutation which occurs by this method, that is base pair substitution, when an incorrect base is incorporated into DNA. Some base substitutions can occur because purines and pyrimidids, they exist in two different structural forms. See the figure here. This is a purine and a pyrimidine. The most common form, it results in the base pairing between adenine and thymine and between guanine and cytosine. You know this very well. This is the normal Watson and Crick pairing. But the hydrogen atoms can move from one base with altered hydrogen bonding property, causing a tautomeric shift. When a tautomeric shift occurs in adenine and the adenine loses its capacity to bind with thymine, but instead it binds with cytosine. This is the molecular explanation for base pair substitution. So the hydrogen atoms, they move from a base with altered hydrogen bonding properties. This shift of hydrogen is called as a tautomeric shift. When a tautomeric shift occurs in adenine, the adenine can now bond to cytosine. And a tautomeric shift in thymine, it allows it to bind with guanine with 3 hydrogen bonds. This error which is there in DNA replication is passed on to the cell's progeny. Next generation, the change is passed. So what happens, the AT base pair which was there Due to this tautomeric shift and changing of the bases, AT base pair eventually will get converted into GC base pairing, as you can see here in the animation. The change in the single nucleotide in the DNA, it results in the change in the corresponding nucleotide in the messenger RNA, and this alters the protein. This change in the codon can result in a different amino acid being incorporated into the protein. So you can see here the mutant arginine, it is a not as that the AT base pairing in the wild type, which was responsible for lysine on the mRNA translation, has got converted into GC. So there is a change. Now this base pair substitution, it can be point, missense, nonsense, whatever it is, and it has a marked effect on the phenotype. It may or may not have, depending upon the type of mutation. Now just let's see all these three: addition, deletion and base pair substitution from your perspective. I want you to note down the amino acid and the codon and the amino acid when I show you the example, and you will get an idea immediately in my next animation how this affects the protein synthesis. So in this example here we have a table showing the DNA codes and the corresponding amino acids. So this is the third pair, this is the first pair, and these four combinations, these are the second pair. Means if this is A, let us say here, if this is A will be phenylalanine, ACA will be cysteine. You take any random, this G, C and T, G, T and C, this is G, T and C will be glycine. So this is the table showing genetic code corresponding to amino acid. Now let us see that three types of mutations which are there. So the first which is there, this is a mutation which is a change in the sequence of bases. You know very well. Let's look at the effect of some mutations here. At the top, which is shown in blue, is a sequence of bases from the coding strand as a part of a gene. So this is a gene. So I want you to just write down the base sequences of the amino acid for which it codes for. Say AGT. So you have A, G and here T. Okay, A, G and T. This will be coding for serine. See here now. So we have AGT coding for serine. So write it down, AGT serine. We have only 4 codons for example. Next is T, G and C. So you have T, G and C. So threonine. So T, G and threonine. See here. So this is threonine. TGC threonine. Similarly, GAC will be G, A and C will be I reckon proline. See here, G leucine, sorry, GAC will be leucine. So it is like this, and this is DNA normals. CTA will be aspargine or aspartate. So now let us see the effect. The first is substitution. Now let's look at the effect of some mutations. We'll start by looking at what happens when we change one of the 3 bases for another. This is called as substitution. If you select one of the individual bases in turn to see what happens when it is changed for another, you will have to wait for one of the reset before changing. Write it down, use the information, and you can just keep in your notebooks. Now see, I will just change G. I will change. This is substitution. Let's AGT substitute G for A. What has happened? AAT it has become. So only one change. I can change G here. GAC, change the G for C. Now see, it is coding for valine. See in your notebook what it coded for earlier. Okay. If I change now TCG, I change this G to G. It becomes threonine. So there is a change in the sequence of amino acid. That's what I am trying to tell. Now see, we will be seeing deletion. If you delete this base, then it will disappear, and what happens with the reading frame you can see here. I will just delete A. See here, A is deleted. Now instead of AGT, the sequence has become GTT. So now you see your 4 codons, first 3 codons, what are there? Now this is glycine, arginine and tryptophan. Glycine, arginine and tryptophan instead of serine, threonine and leucine, it has become, instead of serine, leucine and threonine, it has now become glycine, arginine and tryptophan. So that is the change which is there, right? So lastly we will see what happens when there is addition of a base. So in this AGT, TGC, GAC, CTA original reading frame, I will add thymine molecule and the sequence will completely shift along to fill the gap and producing a new reading frame. The complete reading frame will be changed. So I will hold this T, put it here. See, the complete reading frame has changed from the original one what you have written down. Now it has become isoleucine, asparagine, alanine and glycine. So this is how mutation affects the protein synthesis, and this is how the addition, deletion and base pair substitutions they function. So that is all for now. Mutations, they are very significant for molecular biology studies. Mutations, they are having a deleterious effect as well as beneficial effects also. The molecular mechanism of mutations are induced mutations and spontaneous mutation. This will be the point of my discussion in the next lecture. Till then, goodbye. Hope you enjoyed this lecture. Study well, try to understand the key concepts. If you have any doubts and difficulties, I am always there to solve it.